Thank you. My name is Jumai Abioye, and I'm the founder and CEO of PanAccess Innovations. Um, we can go to the next slide. PanAccess is pretty much a, my brainchild or a baby in a sense that I decided to start last year when I decided to quit my job or leave my other baby that I was building, not an RNA, because I got to the point where I realized I was building a lot of technology that my parents would never be able to afford. I constantly am looking at avenues to get more medication, more therapeutics, more diagnostics to more people globally. So PanAccess is focused more on how do we develop with a larger set of people in mind and with markets that we wouldn't normally consider as beachhead markets or markets that we wouldn't even think about for commercialization in mind. So we can go to the next slide. We have a small team in Calgary and are supported by a wide range of supporters, including the ADEPT program, which we've been very, very fortunate to have access to, and other players and stakeholders within Alberta and across Canada. Next slide, please. The key context to keep in mind here is that about 85% of people globally live on less than $30 a day. That's a huge amount of money, but that includes your house, car, feeding, every single piece is what that $30 a day means. And that's 85% of the world. So they would not be able to afford tens to hundreds of dollars of tests, which is why they tend to be excluded from the market segmentation that we tend to present. So what does our TAM look like? What does our SAM and SUM look like? We can go through the numbers of people in the world, but actually 85% of those people would never be able to touch this diagnostics or therapeutics. Also, most of these people are of this 6.7 billion people that live in low and middle income countries. There are people in this room who are related to those 6.7 billion people and whose hearts are always pulled towards those 6.7 billion people because Canada is diverse. Last year, after quitting my job and spending a lot of time in economic development data, I realized that the difference between the life expectancy of someone born in Nigeria and someone born in Canada is about 30 years. And so that means 53 years versus 83 years. So when my uncle died earlier this year of sepsis, which still no cause for, at close to 60, it made me reflect that, oh, he was already past his life expectancy. That is not okay. One of the things that can really help in solving this and addressing this is diagnostics. So next slide, please. So there is an affordability gap as we think about the tools that we are building, as we think about the products that we're developing. But we also have issues with overuse of antimicrobials and antibiotics in the agri-food industry. That's where most of our antibiotics go to. So when we think about antimicrobial resistance and solving that, we should be thinking about how do we help guide the usage of these tools. Testing can help with that. We also have health inequities, even in Canada here, with rural and indigenous communities that struggle to be able to access testing, or families that have to drive really long distances to come to major cities to get tested. And so being able to have access to home testing is a pretty critical piece. Yeah, that's a lot of the context, a lot of the information around the problem, and then we can move forward to what does a solution look like, or what are we trying to achieve? Next slide. So the opportunity really is that Canadians and their loved ones need fast, accurate, and reliable tests, whether it's at home, on farms, in schools. We need simple tests that we can use, and usually a platform really lends itself to that. Next slide. So we, to be able to address this and pick something very, very simple that has the impact to the possibility or the potential to have a significant impact, we decided to marry two basic technologies: nucleic acid amplification, which most people are used to with PCR, So if you keep in mind your COVID testing lingo, PCR, and then your classic lateral flow tests, again, the ones you do at home versus the ones that are done in the labs. What if we can bring the precision of PCR to lateral flow tests, and then we can develop really, really cheap tests that can be used at point of need. And this technology is not novel. These tools are not novel. They have been published a lot of times. So what is the challenge in commercialization? It tends to be that you have antibodies from Abcam or from Thermo Fisher or from some of the other bigger players, and you have polymerases from NAB or from other players. And so that lends itself to licensing challenges, IP issues, supply chain issues. And then you also have the question that Michael was speaking about earlier, analytical utility, analytical validity. What exactly is the purpose of this test? So there is a need. There is a technology that can address that really easily, that is a platform. We know that in times of trouble, we've been able to deploy lateral flow testing in really, really large amounts. We know that we've been able to deploy PCR type tests in really, really large amounts. So we should be able to use this for a lot of at-home tests. Our technology is versatile. You can use it for different applications, single temperature operation, so isothermal amplification instead of PCR that requires really expensive equipment. And then a broader detection capability. A lot of nucleic acid lateral flow assays have actually been validated at greater than 95% sensitivity and specificity. So there is a lot of information about the use case for this test. And so the approach that we've taken is to look at the supply chain and go, how do we actually drive this into practical use? Next slide. So the goal is to have a Canadian innovation with a global impact. We produce all of our critical raw materials in-house, our antibodies, our polymerases, all the different components that enable us to be able to integrate or consolidate the supply chain for these tests. And then we eliminate supply chain, regulatory, and IP bottlenecks and reduce the costs to ensure affordability globally and in Canada. And the goal with this is to be able to have a distributed manufacturing model where we can out-license this technology to the low and middle income countries in the future so that they can produce locally and even drive the cost down, but also have their own regional targets and modify this test to what they need to test. And so you have affordable distributed model, and it's scalable and versatile. Next slide. To date, we've completed a lot of the key pieces for making our kits, our devices. We've completed our enzyme engineering and production. We've completed our antibody design and manufacturing, our strips and cassettes to be able to reduce plastic usage and ensure sustainability as you start thinking about increased testing. Consolidation of supply chain on consumables that will go into the kit. So we've developed the core components of our kit so that we can actually validate with the materials that will go into the kit itself, as opposed to showing you validation data on things that we've purchased. We've actually made all of ours. Next slide. And then we've done laboratory evaluation on a range of applications. And these applications are to demonstrate the platform nature of these tools. And so a lot of these laboratory evaluations are done with simulated samples, especially looking at just the region of the genomic DNA that we want to target, and then show we can target that. Our primers work, they're efficient. In silico designs for a number of tools that we need to use. And the broad range of that goes from non-invasive prenatal testing for baby gender to testing for avian influenza or gonorrhea in humans. And so we can also do customizations and partner with people who want to develop their own point of care tests. Next slide. And so When it comes to our intellectual property, it's always very simple for us. It is patent products protect processes. And so our own in-house produced antibodies and polymerases and the kits are our intellectual property. We own them, we design them to match against market. So we have our antibody fragments that we're able to produce recombinantly and eliminate the expensive production processes that will classically be used for antibody manufacturing. And then we have our manufacturing processes and product workflows that we own, and those will be protected as trade secrets. And then there are designs, engineering designs on things like cassettes that are going to be protected as designs. And so we've secured our funding with Elevate IP Alberta to be able to implement our IP strategy and work with law firms that support that. Next slide. Market and competitive landscape. These are all the major players, big billion-dollar market that Michael dispelled a little bit of earlier, but there is a billion-dollar market. We're looking at about an $83 million market in the agri-food industry for the total obtainable market for ourselves, or serviceable obtainable market. Next slide, please. So what are we looking for as an ask? We've built all the critical assets that would go into products. This company is a private company funded through myself and grants. And we're finally getting to the end of our runway. And so we're looking for anchor partners. It's a social impact company. And so it's not lending itself to a classic venture model in terms of the revenue generation. Of course, nobody wants to go to the 84% that has the pennies to spend. And so this is a critical piece where we're looking for anchor partners who are interested in any of these tests that we can either co-develop or develop for them and help them generate tests that can be used on site using our kits. We're looking at collaborations as well as just expanding our IP assets. The joy of this is that now we can out license to low and middle income countries, and they can own their own IP and not need to license anything to be able to produce these types of tests. And so the goal would be to extend this model to other things under the PanAccess umbrella in the long run. Next slide. So we're excited about opportunities. Our initial goal would really be to initiate product delivery to an anchor partner. So that's I guess the battle cry or the hard cry here, which is if you're interested, if you have a test that needs to be developed that has any DNA or RNA target, we can do that. Our platform can do that. We can design the target components, and you can have your strips and your tests, your classic lateral flow tests to be able to deliver that. Next slide, please. And so last slide, which the QR code for contact. Please reach out, get in touch if this is of interest. Thank you.